Welcome to Lakshman Biology. Now we will discuss about dihybrid cross. Dihybrid cross. Dihybrid cross also called as inheritance of two genes. Dihybrid cross. It is also called as inheritance of two genes. Mendel did inheritance of two genes or dihybrid cross to know about how the genes, if there is more than one gene, how that genes are inherited from one generation to another generation. And after completion of dihybrid cross, he identified that when the more than one gene is inherited from one generation to another generation, one gene inheritance not depend on another gene inheritance. He explained that in the law of independent assortment. What he identified by the dihybrid cross? He identified that when more than one gene is inherited from one generation to another generation, the transfer of one gene from one generation to another generation not depend on transfer or inheritance of another gene. For this, Mendel taken two genes. One gene is responsible for the seed shape. One gene is responsible for seed shape. Another gene is responsible for seed color. Dihybrid cross, if you see the definition of dihybrid cross between two pure lines. Cross between two pure lines which differ in two characters. That is called as dihybrid cross. Okay. If you see the genotype of parent generation, the genotype of parent generation is homozygous dominant. It has homozygous dominant genotype for seed shape, homozygous dominant genotype for seed color. Capital R, this is the dominant alleles that will give dominant characters round. Round seed shape is dominant over the wrinkled seed shape. Yellow color is dominant over the green color. Then if you see the homozygous dominant alleles or homozygous dominant genotype of seed color is capital Y and capital Y, that will give the dominant color, that is called yellow. When you cross the round yellow seed plant with wrinkled green plant, this wrinkled green seed plant contain the genotype that is homozygous recessive for both characters. Smaller will give wrinkled seed, that is wrinkled is recessive over the round seed. Next, small y is responsible for the gene green color seed. Green is recessive over the yellow color. Then, during the formation of gametes in the two alleles, only one allele is entered into the gamete. In the both two capital R's, only one capital R enter into gamete. These are the gamete. Here also, during formation of one small r, one capital Y enter into the gametes. These are the gametes. During the formation of F1 generation, these gametes are fused together. Hence, here one capital R from one gamete, one small r from another gamete leads to formation of heterozygous genotype for the seed shape, and one capital Y from the one gamete, one small y from another gamete leads to transfer of heterozygous dominant for seed color. That is capital R, capital Y, small y. This is the heterozygous dominant genotype for both characters. Then when we selfing means crossing of plants which are present in one generation found from the same parents. Same generation plants if you cross, that is called as selfing. Like this is the one plant which contain heterozygous dominant genotype, and it is the another plant from the same generation which contain heterozygous dominant genotype. When you cross these two plants, we can see the formation of F2 generation. First, how the gametes are formed, that we will see. Both one organism, we can see how it will form the gametes. During formation of gametes, again, only one allele in the pair is enter into gamete, capital R. When capital R from the heterozygous dominant genotype of capital R and small capital Y, both are combined together, that leads to formation of gamete which contain capital R, capital Y. When in case if small r and capital Y are joined, we can see the formation of smaller. This is the second type of gamete, small r with the capital Y. Next, in case if capital R joins with the capital, if capital R joins with the small y, that leads to formation of gamete with one dominant allele, one recessive allele. When small r is joins with the small y, then you can see the formation of gamete with two recessive allele. These are the four gametes. Four gametes can be formed from heterozygous dominant plant of F1 generation. Similar type of gametes are formed from the both parents. Now, if you observe the possible F2 generation plants by the Punnett square, this is the Punnett square. See, this is the first gamete, second gamete, third gamete, fourth gamete of one parent. Same, first gamete, second gamete, third gamete, and fourth gamete of another pair. If these two gametes are fused together, you can see the formation of homozygous dominant genotype. Then this gamete and this gamete. Okay, in the same line you have to see. We can see the formation of heterozygous dominant genotype. Finally, we can see the 16 different variety of genotypes in F2 generation because of 4 different types of gametes of each parent. That if you observe the genotypic ratio that we can observe, the genotypic ratio is genotypic ratio of F2 generation. Genotypic ratio. Total 16 plants are formed. 16 different genotypes are formed. Those genotypes are in the ratio 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. Then what is the 1 here? 1 is the homozygous dominant. This is the 1. Here I write. This is the 1, 2. 1 is to 2 is to again 1. Smaller, capital Y. 1 is to 2 is to 1 is to, then this is the 2. 2 is to, these are 4. Heterozygous dominant plants. 4 is to, then again 2. Sorry, 1. This is the 1. 1 is to 2 is to 1 is to 2 is to 1 is to 2 is to 4 is to 2. 2 is to 1 is to again 2 is to 1. This is the genotype. 1 capital R, capital Y, next 2 capital R, small r, capital Y, capital Y. Next 3, 1 is to 2 is to 1. Next 1 small r, capital Y, 1 is to 2 is to 1 is to, next 2 capital R, capital Y, small y, 2 is to 4. 4 here. There is a 4 genotypes, 4 heterozygous genotypes. 2 is to 4 is to again 2. Capital R, sorry, again 2, small r, capital Y, small y. 2 is to 4 is to 2 is to again 1. 1 capital R, small y, small y. 1 is to 2 capital R, small y, small y. 2 is to 1. That is small r, small y, small y. That is the genotype. If you observe the phenotypic ratio, genotypic ratio 1 is to 2 is to 1, 2 is to 4 is to 2 is to 1 is to 2 is to 1. Then if you observe the phenotypic ratio, that is 9 is to 3 is to 3 is to 1. Here 9 round yellow seeds, 3 round green seeds, 3 wrinkled yellow seeds, 1 wrinkled green seeds. That is about the dihybrid cross of Mendel. After the dihybrid cross experiment, he identified law of independent assortment. In next class, we will see what is the law of independent assortment. Thank you.